Hopefully everyone can see me and hear me okay. My name is Jill. I am a guide leader in Birmingham with the Maypole Guides, and I've also been a guide leader in London and a ranger leader in London. And I think you can tell from my accent that I'm not from Birmingham. I'm actually from Canada. And I used to be a guide leader in Canada, and I went through guides as a girl in Canada as well. So I've been doing lots of things with guides for a long time. And I'm also a scientist. So I work at Aston University, where I run a research lab and I teach university students all about biology. But today we are going to be doing a fun activity with DNA. So this is some DNA origami that I've made from a piece of paper. So hopefully everyone has their DNA origami template printed out, and we're going to go through this together. So if everyone can take their DNA origami template and start cutting off the white edges. So you just need a pair of scissors. You need to cut along this outside line at the bottom, this other one at the side, and just imagine that there's a dark line along the top of the template there. So if everyone can take the time to cut out their template, I'm going to go ahead and share my screen so you can see the instructions of what we're going to do. So I'm just going to hopefully make that big enough so everyone can see it. So I'm going to do this together with you. So I'm going to cut out the edges of my template. And if you have any questions, please put them on the chat in YouTube. And my friend Rebecca is taking a look at the chat, and she's going to make sure that if you have any questions, that she will relay those questions to me, and hopefully we can answer them. So I'm just finishing with the cutting of the edges off my template here. All right, and it's best if you can do this on a flat surface like a table, because you need to make some pretty firm folds on your template. So I have my template now that has the edges cut off. So all I'm going to do is fold it in half along that heavy black line right in the middle of the template. So make sure it's as even as you can get it, and just make a nice strong fold. Maybe press it down with your fingernail and make sure you have that one piece of paper that's now folded in half. So hopefully you can see that the letters are only on one side, and they're only on one side of the reverse as well. So at this point, it's really important to make sure we have the right side facing up. So we have now just done step one here on our instructions. So we have folded in half lengthwise and make those creases as firm as possible with your fingernails. Okay, so now we're going to hold the paper so that the thick lines are diagonal and the thin lines are horizontal. So that's this side. I'm going to hold that up to the camera. So you want to be looking at the side in which the diagonal lines are thick and the horizontal, the straight lines, are thin. So you want to have that side facing you. You might want to lay it down on the table. So when we're making these folds, we need to make sure we have the thin line on the inside of the fold and the thick line on the outside of the fold. And this is my cat, Darwin, who really likes to be in Zoom meetings. So what we're going to do is hold the paper so that the thick lines are diagonal, the thin lines are horizontal, and then we're going to fold the top segment down. So you could, if you're looking at it from your perspective, you can have it flat on the table. But as you can see, I'm just folding that first segment down so that I can see the thick line on the back side of it, and the thin horizontal line is on the inside. So I'm just going to repeat that for every segment going down. So I'm going to fold it, make sure it's a nice strong crease. I'm going to use my fingernails and then unfold it. So I've got the first one folded down, I've got the second one folded down, and now I'm going to do the third one. So hopefully you're all keeping up with me and you're making your folds going all the way down your DNA origami. So making sure that the thin lines are on the inside and the thick lines are on the outside. And as you can see, Darwin is very interested in what's going on here. There you go, Darwin. Yeah, you're being a very good cat. You're gonna have to wait though if you want a treat. You're gonna have to wait until we're done doing what we're doing here with our DNA origami. So hopefully everyone's still with me. As you can see, I'm doing this sort of just holding it up in the air and using my fingers, my fingernails to make those creases. And Darwin is continuing to help me with this. Excuse me, Darwin, thank you very much. No, no one wants to see that. Okay, there we go. All right. I only have 2 more folds to go. I should have closed the window and kept him outside. I didn't think my cat would be participating in our DNA origami session today. Okay, so I have now made my last horizontal fold. So I have gone all the way down the template. Thank you very much, Mr. Cat. All the way down the template and just quickly making sure that I have all of those folds that have been made for every section of that template. Thank you. Thank you for checking. All right, so remember, we had facing us, the diagonal lines were thick and the horizontal lines were thin. And now we're going to flip it over onto the other side. So when it's facing you, if you're laying it on the table, when it's facing you, it should be curled going under like this. Sorry, excuse me, Mr. Cat. All right, so you should have a curled piece of paper like that, and it should be facing down. So we are now working on, that was step number 5, turning the paper over. So hopefully everyone can still see that. So we've turned the paper over, and now we're going to fold along the first diagonal line. So we've made all of our horizontal folds on one side. There we go, all of our horizontal folds on one side, and we're going to make our diagonal folds on the other side. Thank you. Can you just sit there? Thank you. I'll get to you in a second. So again, you can either do it laying flat on the table, or if you want to hold it up in the air like this. These ones are a little trickier. So if you're small and you need some help, hopefully you've got a grown-up there who's able to help you. So you want to basically be careful that you're starting the fold at the corner where the colored bit meets the white bit. So you just want to focus your first bit of that fold on the corner, and then use your fingernails and make that fold the rest of the way. So now I have my diagonal folds facing the other way. And same basic principle, you're just going to do that all the way down your template. So hopefully everyone is doing that. So you make the fold, first segment down on the diagonal line, second segment down on the diagonal line, and you just keep making those folds all the way down your template. And hopefully my cat is not being too distracting for everyone. All right, what I think I need to do is give the cat some treats, because he's probably not going to leave us alone until I do this. So there you go. There you go. He has a nice pile of his favorite treats. I'm sorry that he just put his rear end in the camera. Okay. Fun with cats. It's part of biology, right? So let's continue making our DNA origami. So remember, we're doing the horizontal folds. So every horizontal fold all the way down the paper needs to get folded. And hopefully by the time you're about halfway down, you'll see how the structure is starting to twist. So remember, when you're making these folds, that the thick line is on the outside and the thin line is on the inside. So it's the same basic idea as what you did for the horizontal folds, only now you're just doing these at a slightly different angle, and you're following those horizontal lines all the way down your origami template. All right, so hopefully everyone's with me. I haven't heard any questions coming through from YouTube yet, so hopefully everything's going well on that end of things. I've only got 2 more folds to do. The cat has been satisfied with his treats and is now sitting in the window and not blocking the camera. So there you go. You can tell that I'm a biologist because I named my cat Darwin. All right, so now hopefully you can see that the model is starting to twist. Hopefully everyone can see that. So we had our horizontal folds on one side, making it kind of like a C shape. And now that we have our diagonal folds on the other side, hopefully you can see it's making a twisting pattern. Okay, so hopefully everyone's at that spot right now. That is step number 9. So let's move these instructions up. So now what we need to do is slowly make every one of these folds, but kind of combine them together into one solid piece. So you're just going to go back and forth. So hopefully you can see that, what I'm starting to do. So it kind of looks like a bow tie at first, and then you're just going to go back and forth with every one of these folds, making sure you're doing the horizontal one way and the diagonal the other way. Okay, so there we go. Oh yeah, I forgot a step. I forgot step number 7. Sorry about that. We need to fold the white edge without the letters up. So we need to fold the white edge without the letters. That needs to be folded up so you can see the letters. Sorry about that, everyone. I got ahead of myself. So I was maybe distracted by the cat. And you want to do that on both sides. So you want to fold those white edges so you can't see the white edges, and all you can see is the letters on the edge. Okay, so it ends up sort of like this. And then you want to unfold them part way so that you sort of have a right angle. So it's like a square edge, part of it's flat, where the color is flat, and then the white edges sort of stick out at right angles. And then you can start doing your other folding. I was wondering why it felt a little strange. There we go. So now all we need to do is just keep those folds that we made before and go back and forth like this. So hopefully everyone can see that okay. And as you do that, you can see that it starts to look like a spiral staircase. So hopefully you can see that nice spiral formation that's coming up. The structure of DNA is what we call a double helix. A helix means it's a spiral formation, and a double helix means that it has 2 sides. So the edges of the DNA structure are alternating sugars and phosphates that are really tightly bound together that make a nice firm outside of the DNA structure. So the sides of the ladder are nice and firm and stuck well together, whereas all of the base pairs, so all of these A, T, C, and G, that is where the genetic material is carried. So what you have is a sugar and phosphate backbone on both sides of the DNA molecule that keeps everything held together. And then you have these DNA base pairs in the center that when the DNA needs to replicate, it just unzips like a zipper down the center, and then you can make an exact copy of this DNA. And if you're interested, if I move on to my next slide, you can actually decode this DNA. So DNA is like a book. And like the books you get at the library or the ones you have on your bookshelves, all those books are made out of letters, and the letters are made into words. And those are the words that we can understand that give us a code. So with these DNA letters, these A, T, C, and G, they are read in groups of 3. So every 3 letters is one DNA word, and that word is translated into another molecule that we call an amino acid. And those amino acids, you can think of them like different types of Lego pieces. They may be different colors, they may be different sizes, but when you put those together, they make a protein. And protein is what pretty much all of our bodies are made out of. So your skin has protein in it, your bones are made out of protein, your hair is made out of protein, and proteins are what keeps our body going and keeps all of the messages zipping around our bodies and allow our cells to function. So if you want to, you can translate the letters on your DNA, and I'll show you how to do the first one. So the first one I have here, hopefully you can see that, is CCT. So what we want to look up is first position here on the left is a C. My second position is also a C, so see how it goes up at the top there. Second position is C, so I'm reading from the left, C, C, and then my third position is T, and that one, it doesn't look like it actually exists, so that's interesting. Let's try the other way around. Let's see what we have on the other side. We've got GAG, so that might be a little bit more useful. So G, A as the second one, and G is the third one, and that is an amino acid called glutamic acid. And yeah, that's one of the 20 amino acids that make up all of the proteins in our body. So there you go. Now you can translate your own DNA. Hopefully you've enjoyed this little activity. Hopefully you have fun with your DNA structure. And I hope that worked out for everyone. And yeah, have fun making your DNA and go ahead and try to translate all of those 3-letter codes you have in your DNA. I think one side might work out a little bit better than the other. There you go. Enjoy. Have a good day. And hopefully I will see you all at 1:30 p.m. or 2 p.m. depending on if you are a rainbow or a brownie or a guide or a ranger. And we are going to do an activity called Make a Microbe. And that's all about good kinds of microorganisms that help us do some really interesting things. So I hope you enjoyed this activity. Have fun with your DNA and enjoy the rest of Fascinate.